The nucleic acid RNA plays a big role in both transcription and translation. So let's take a look at RNA. Now, there are three types of RNA. The one that's going to play the biggest role, or at least the most visible role in what we're talking about, is called messenger RNA, and it's referred to as mRNA, M for messenger. Messenger RNA is a temporary copy of a gene. In other words, it's the index card that we talked about in the previous video. The next kind of RNA is transfer RNA, or tRNA. Transfer RNA carries an amino acid to the ribosome, and it's actually going to be our translator. One side of the tRNA is going to have a code that matches the mRNA, and the other side will have the appropriate amino acid. The third kind of RNA is ribosomal RNA, and it is actually part of the ribosome. It helps to read the sequence on the mRNA and match it to the appropriate sequence on the tRNA. Now, compared to DNA, RNA is short. The longest RNA would be messenger RNA, and it's only one gene long, whereas DNA is thousands of genes long. RNA is single-stranded, unlike DNA, which is double-stranded. Because it's single-stranded, it's also less stable. That's what makes it a temporary copy of the gene. Unlike DNA, RNA uses the sugar ribose. Remember, each nucleotide has a sugar and a phosphate and then a nitrogenous base. In DNA, that sugar is called deoxyribose. In RNA, it's ribose. And that sugar is what helps each enzyme to determine which nucleotide is appropriate. Am I making RNA? Then I need to look for a nucleotide that's made with ribose. Am I making DNA? Then I'll look for a nucleotide made with deoxyribose. So the next thing that's different about RNA from DNA is that instead of thymine, RNA uses a nucleotide called uracil. So everywhere that we would put a thymine, which means across from every adenosine, we're now going to put uracil. And finally, RNA is made by RNA polymerase, not DNA polymerase. RNA polymerase looks for the sugar ribose and puts the appropriate nucleotide in the new molecule of RNA.